Good morning and good afternoon, everyone, and thank you for joining me today. As Antonio mentioned, we will be doing an introductory webinar into Ingenuity variant analysis. And for those of you who were with us for the last hour, you heard all about analyzing your gene sequence data using CLC Genomics Workbench. And now we will be talking about how you can interpret that data using biological knowledge in Ingenuity variant analysis, which is the last piece of the QIAGEN sample to insight pipeline. So before we get started, I wanted to give you a little background into the application and tell you a little bit about it if you're unfamiliar with this platform. It is an intuitive, user-friendly web interface that does not require any bioinformatics skills, and it provides an interactive filter cascade that can enable you to easily eliminate or keep variants using a basic set of filters, and you can do your analyses iteratively. So if you have different hypotheses in mind, different genetic models that you'd like to test, you can visualize, evaluate the results in real time, and then reapply a different set of filters to interrogate other questions. Ingenuity variant analysis also provides access to our knowledge base, which is a large collection of expertly curated and accurately updated information from peer-reviewed literature and databases, and this includes millions of gene and disease interaction findings as well as mutation reports. And we allow you to conduct causal network analytics to find novel genes that may interact with known genes for a given disease or pathway that you might be interested in. This tool is designed for sharing your data and information with others, so you can share it more privately with your colleagues and collaborators within a group, or you can share your analysis more publicly as a supplement to your paper by using our publication tool in the application. In addition, this is designed for scalability, so we're enabling analysis of thousands of genomes and whole exomes simultaneously. This is a little quick snapshot into the knowledge-based content that is available through variant analysis. We have over 10 million findings curated from peer-reviewed literature as well as publicly available data sources. About 4 million of these are mutation-specific findings where you can know whether a particular variant has been previously associated to either increase risk or cause a disease. And then we have built-in disease models that are also curated into the knowledge base. For us, a quick look at the publicly available resources, here are some of the more commonly used ones like Cosmic TCGA, OMIM. We also provide in silico tools such as SIFT and Polyfen. And now, as BioBase is also part of the QIAGEN family, we provide access to HGMD for variants in your data set, so you can get more information about what is curated in that database as well. So for the purposes of today's webinar, what we will do is we will talk about how you will manage your samples in the application to upload, annotate, and search samples. And then we'll look at setting up an analysis, take a little deeper dive into the filter cascade, and also review the results in the analysis, in the application. For those of you who are familiar with variant analysis, you may be using it for one or more types of different case studies or analyses. This is a quick overview of the more commonly used case studies in the application where people have small or large families and they're looking for inherited disease mutations or causal variants. People also have small case control groups. These are unrelated cases where they want to compare them to a group of unrelated controls and see if there are any commonly mutated genes in the case samples. In addition, you may have a large data set of cases or controls or variable phenotypes, so a quantitative trait, and you would like to see if there is a rare variant burden associated with the disease trait that you're interested in. And you can do that using our statistical association tools in the application. And also look for tumor-specific variants. You can filter out any germline variants from your tumor samples and interrogate those variants further for biological relevance and also pharmacogenetic finding. And for the purposes of today, I will use a somatic tumor normal comparison analysis to go over the filters as well as the analysis review. However, if you have any of the other types of case studies and you'd like to learn more, please call our support and we will guide you through those as well. So starting out in an application, when you log in, you will see the analysis view. But what you want to do first off, obviously, is to upload your samples. So when you log into Ingenuity Variant Analysis and you'd like to upload samples, you would go into the Samples tab and click on the Upload option. This will launch a window that will either allow you to upload your samples one at a time. So if you have just like a couple of VCF files for a few samples, you can just go ahead and use our direct interface to upload those samples. However, most people have large data sets with a large number of samples, and a lot of times these samples are in a multi-sample VCF. For that, we would like you to use our data stream, where you can click on the link for the data stream uploader and then just drag and drop your VCF file into this window, upload it. The file gets validated in the back end and is uploaded into your account, and you get a notification that your samples are now available for use. So if you have an active subscription, samples are uploaded into your account, you can just start analyzing them by setting up an analysis that we'll talk about in a minute. However, for some types of studies, you may need to annotate your samples further. So to do that, we have an annotate option available in the application. So first off, if you were doing a matched tumor normal analysis like the one that we'll see today, you need to add subject identifiers for those tumor as well as normal samples. And this is the same subject ID for the tumor and the normal. This way the system knows that they're from the same individual. So to do that, you click Subject Identifier and use a tab delimited text file that can be created using Excel, for example, and set up your subject ID. So you can see here, there's the display name. This is the name that you assign to each of your samples in your VCF file. And then you will use a subject ID that will designate both the tumor and the normal sample as belonging to the same individual. Once you've done that, you upload the file into the system. The application will show you how many of your samples were annotated. And this is helpful to determine if all the samples that you included in your text file were appropriately annotated. So if you have 10 samples and it's only showing 6, you probably have some kind of a mismatch between the display name in your subject identifier file and the system, and you can check that. Next up, if you were trying to analyze large pedigrees and you'd like to annotate the samples and put in the relationships for parents to look for transmission of the causal variants, you can do that using our pedigree annotation options. Click on pedigree and upload a PED file. A PED file is a 6-column file that's similar to a PLINK file format, where you have the family ID, individual ID, paternal maternal ID, sex of each of the individual, and the phenotype, whether they're affected or not. Phenotype is not required, but it's just nice to have it, so you can then search samples using that information. Once you upload your PED file, you will also see how many samples are annotated, and then you can proceed if it seems like all samples are appropriately assigned to their respective parents. Next up, if you are doing quantitative trait analysis, or if you just wanted to add any clinical features to your samples, you can use our clinical features tab. And when you click on that, you can upload a file with the subject IDs of all the samples in your account and whatever phenotypes that or clinical features that you would like to annotate. So I'm just showing you a simple 2-column file here. It's a subject identifier and a generic phenotype for case and control, but this could be as elaborate as lab reports, so cholesterol levels, enzyme levels, or anything else that you would like to include for each of your samples. Once you have uploaded this file, once again, you'll see a display board showing you all the samples that were annotated, what different phenotypes or criteria were associated with those samples. So once again, you can check to make sure all your samples are appropriately annotated before you proceed. Once you've annotated your samples, when you click on any of the sample names in the sample information panel, you will start to see a list of all the clinical features that you have associated with the given sample listed right at the bottom of the general information about the sample. From this point on, you can either click on any of these gray links to select samples that all share the same phenotype. So if I were to click case here, I'll see all the samples that have been designated as case in my data set. Or if I wanted to search for samples with a specific phenotype, in my samples page, I can just type that phenotype header. So in this case, it's very generic. It's just phenotype equals case. But you could have, say, cholesterol equals 100, and then every sample that has a cholesterol of 100 will show up in your list. So here it's showing me 29 samples where the phenotype is case. This is helpful when you're setting up an analysis. You can select specific samples for a given phenotype. If you wanted to select samples that just have a given phenotype, if you just type the word phenotype, it will list all the samples that have a phenotype annotations, whether they're case or control. And this also helps you to select all the samples that have a particular clinical feature that you would like to include in your analysis. So that's all about managing your samples in the application. I will now move on to setting up an analysis and then talk about the filters and review the analyses. If you have questions, I think it would be best if we take them at the end. So please note your questions about sample management. And now we're going to talk about setting up an analysis. So to set up an analysis, I'm going to show you the way to do it from the My Analysis page, where you click on Create Analysis, and you get a pop-up window or a page that asks you to start defining the criteria for your analysis. So for example, you obviously need a unique identifier for the analysis, so you can type a name. We only allow unique names for analyses, meaning if an analysis but the same name exists, you will be required to add some unique identifiers to make sure that those two analyses are not confused. And then you will pick your study model. So if you're looking for an inheritance model, or if you're looking to look for somatic variants, you'll look for cancer. If you had a large number of cases controlled, then you need to do statistical association. You'll do a stratification study. If you're just looking at a single genome and want to see what's in there, you can start as a personal genome. However, it must be noted that this option is going away in our summer release, so you will no longer have a personal genome option. However, if you had a single sample and you select genetic disease, you'll be able to set up an analysis for a single case sample. You can also set up analyses using previously set analyses. So if you have a filter cascade that you've defined for one family and you have another family and you'd like to apply the same criteria, you can just go in to use settings from analysis, find the analysis name, and then it will appropriately set your filters for that. Once you've selected the type of study, the next thing you'll be asked to do is identify the biological context of the study. This is not always required, but it is a feature that if you add some phenotype data or information, it just helps you to narrow down biologically relevant variants in your analysis. In this example, we are starting with a cancer study, so it's asking me to pick a cancer subtype. I can leave it as any cancer and then it will cover everything, or I could pick a specific type of cancer and limit my variance associated with that particular disease. And then I can go in and select my case and control samples here. So in this is where the clinical feature annotation comes in really handy. If you had added some unique identifiers for your specific samples, you can easily search from them by typing those keywords, and then you can distribute your cases and controls appropriately instead of having to search for them in this, especially if you have a large number of samples. Once you have selected your sample, you've set up an analysis criteria, you've given it a name, the next page asks you if you would like to do some pre-filtering. This is really helpful if you have a large number of whole genome files. It will help to upload your analysis faster because it will limit the number of variants that are included. So you can choose to only include exonic variants or eliminate common variants even before they are set up in the analysis, or remove low-quality variants before they get included in the analysis. One thing to note is anything you remove at this point will be excluded from the analysis throughout the setup. And if you want to include those variants, you would have to go back and set a new analysis without selecting those criteria. So in the end, you'll get a summary page that will tell you all about your analysis, the name that you've given, how many samples you have, how many cases controls, if you used any custom annotations, and then you can proceed to set up the analysis by clicking Analyze. When the analysis is done running and is active and available in your account, you'll get an email. And in the email, you can click on the link, be directly taken into your account. You log in and you can see your analysis, or you can log into your account and just go into the analysis tab and click to open the analysis. When you open your analysis, this is the view that you will see in your analysis page. Here on the left-hand side is the filter cascade that was set up for you using some predefined criteria that you had set up in your wizard and setup wizard. In the middle, we have the variant table that shows the position and more information about each of the variants that are included from your data set. And on the right-hand side is more information about a selected specific variant. So for example, here I have this mTOR variant selected, and I can look at more details about that variant on the right. And we will review these details in further slides. So you open up an analysis, and now you want to understand what's happening in the analysis, what filters have been applied. And more likely than not, when you set up an analysis, you'll start with the confidence filter. The confidence filter is available to you to remove variants that have low quality or are otherwise considered suspicious because either they didn't pass the upstream pipeline when you were calling the variants, or they are in highly exonically variable regions of the genome and therefore likely to be false positive. So you can set a criteria for the call quality. You can check to only include variants that pass the upstream pipeline. You can also filter variants using read depth and allele fraction, and then also remove variants that fall in the topmost exonically variable regions in 100 base pair windows. Or you can remove the most exonically variable genes. So these will include genes like mucins, HLAs, titin, any number of genes that have been known to show a lot of variability, and these variants were not validated when Sanger sequenced. So once you have applied a confidence filter, you go down to only variants that are you can trust in terms of quality. And then we go on to remove common variants, because very likely that if you're looking for a causal variant, it will not be something that's too common in the population. And the threshold for how common or rare you would like the variant to be depends on how common or rare the disease that you're looking at is. So here are the different databases that we use: the 1000 Genomes Project, the NHLBI dataset, also the Complete Genome 69 samples that were publicly available. However, moving forward, as of August, we will incorporate this data into our allele frequency dataset, and the option for complete genomics will no longer be separately available. The last option here is our allele frequency community dataset, and this is a dataset that's unique to Ingenuity QIAGEN products and currently available through Ingenuity Variant Analysis. It is a free community resource that's hosted by QIAGEN. You can join the community by going to the website, or if you click on this link here, you'll be able to join the community. This consists of genomes and exomes from our users who have anonymously contributed them into a pool data set where we're now calculating variant frequencies among all of those samples. Currently, we have over 100,000 genomes and exomes, and these represent over 100 countries of origin. There are nearly 14,000 whole genomes that cover intronic regions and splice sites. And this resource, as I said, is free to use, but you need to opt in to share your data with the community to get the data back from the community. So if you have selected to opt into the allele frequency, you will be able to use that option to also filter common variants. Later on in the year, we will break up the allele frequency data by ethnicity, so you will be able to filter using ethnicity-specific common variants. So now that we've removed the common variants, we move on and would like to look at variants that may have a deleterious effect on the protein function or gene expression. And for this, we have a filter called the predicted deleterious filter. This filter, as you can see, is broken up into 3 sections. All options applied here are OR, so if you pick one or more, if one or more of those criteria are met, the variant is retained or excluded, depending on whether you say keep or exclude in this filter. So up top here, we're looking at variants that have been previously experimentally associated with the disease. So these are published known mutations. And then we're using the ACMG guidelines to classify these known variants as pathogenic, likely pathogenic, variants of uncertain significance, benign or likely benign, as well as include variants that are present in HGMD. The second section here is looking to see if a variant is likely to cause gain of function. And once again, for this, we're looking at published literature variants. So if there's a known gain of function variant and it's present in your data set, it will be kept. In addition, we apply some algorithms that are our own Ingenuity algorithms and include BSIFT to also find variants that may cause gain of function, and then look for microRNA binding site variants as well as copy number gains or gene fusion if the structural variant data is available from your uploaded VCF files. In addition, we use a wide array of in silico tools to determine if a variant may cause loss of function. So we're looking at missense predicted to be damaging by SIFT and polyphen, splice site losses. We're using an algorithm called max and scan to find other splice variants, also looking to see if there is a frame shift or start or stop loss or gain. So you can select any one or more of these criteria to determine whether you would like to consider that variant as damaging or interesting for your study. We also have in silico tools to determine tools and databases to determine non-coding variants of interest. So you can look for promoter losses or ENCODE transcription factor binding sites, enhancers, and also further down you can look at UTRs and so on. So once you've selected your criteria for deleterious variants, you can now go on to add other filters. The study that I'm showing you today, we are comparing 2 tumors with their matched normal. So in this particular example, we're also applying a cancer driver filter. The cancer driver filter is designed to specifically interrogate variants in genes that are associated with cancers. So here we're looking at published mouse knockout models of gene associations from cellular processes, pathways, other types of associations from therapeutic targets, also including any variants that are in cosmic and TCGA. And once again, in this filter, you can be as inclusive or exclusive depending on what you are interested in. For most studies, you would also apply a genetic analysis filter. So a genetic analysis filter is used to keep and exclude variants based on the specific question or genetic model that you're testing. So in this example, we'd like to keep variants that are only present in the tumor DNA sequence but not present in the germline. So we keep specific genotypes in the tumor and exclude all variants from the matched normal. To do that, we had assigned samples from the same individual using subject IDs earlier, so now we can pair match them. So this way, it will only remove a germline variant if it's present in the matched normal. And similarly, you can set up genetic inheritance models. So you could have a trio with mother, father, child, and you're looking for either a transmitted variant or a de novo variant. And you can do that using by restricting to transmitted or de novo. If you supply the pedigree information, that will be enabled in the application as well. And if you're more interested in looking at some of those types of data, please call us and we will walk you through that. Now here is the most unique and special feature of variant analysis, which is our biological context filter. This is a filter that allows you exclusive access to the Ingenuity knowledge base where we have all those curated findings from literature and publicly available resources. And so this is really bringing all that knowledge at your fingertips, and by typing the name of the disease that you're interested in or a pathway if you think that's involved, or let's say you have a list of phenotypes for a genetic odyssey case, you can just type those in and then start by looking at genes that are directly associated with that particular disease or pathway or phenotype that may have variants in your data. Or you can start to use our causal analytics by selecting one or two hops upstream or downstream of known genes associated to a disease or pathway and start to find novel genes with variants that may be involved in whatever phenotype, disease, pathway, function that you are looking at. So here, we really encourage you to use the biological context filter and try to narrow down the list of variants that are biologically relevant. In addition, for a number of tumor studies, you may be interested in finding if there are any drugs or therapeutic targets associated with your data set. So you can select the pharmacogenetic filter and then try to find variants in genes that may be associated to response, metabolism, toxicity, or maybe known targets for a particular drug and other finding information. You can do this more generically by including all drugs, as I did for this particular example, or if you want to know if a specific drug has any known targets for response or toxicity in your data set, you can type in the name of the drug and you will get more information on that. So this is the set of filters that I use for the particular analysis that I will go over in a minute. This is the result that you get from your analysis. We looked at this page earlier. On the left is your filter cascade. In the middle is the variant table, and then more details about each of the given variants. So if I select a variant, this panel pops up on the right-hand side, and I can start to go into more details about the variants. More details will give you quality information and overview of the variant itself in terms of where it's located in the gene, what samples carry the variant, what the QC is, etc. And then what you're really interested in is how is this variant associated to the disease or phenotype that I'm interested in. So you click on path to phenotype here, and this window will pop up, and this will show you gene disease association. So in this case, I had not selected any hops, so I'm just seeing direct associations between breast cancer and tumors and the genes listed in my table. By clicking on this link here, I can start to get more information about that relationship from published literature. So there'll be a list of findings here as you scroll down, where you can click on those links to go to relevant databases or to PubMed to read an abstract for the paper, access more information. If I had selected one or two hops and I had some novel variants that were not previously directly linked, I would see those genes connected to known genes and then connected to the disease. And similarly, by clicking on the connection links, I can find more information about those relationships and make informed decisions whether I'm now looking at a novel target, a novel gene that might be involved in this particular disease pathway, or if it's just a spurious association because this gene interacts with everything. And if you're interested in the pharmacogenetics finding, you will click on the pharmacogenetics tab, and you'll get all the information that we've curated in relation to drug response or toxicity or interaction for all given genes and variants in your table. And so in this case, we're looking for drug responses for mTOR variant that we were looking at, and there's the whole list of findings. We're also have now incorporated the PGMD database. So starting end of August with the new release, you'll also see information for PGMD incorporated into your pharmacogenetic findings. And by clicking on these links, you can go and read the primary publications. But also by clicking on a specific drug name here, you can go into the knowledge base and read more about that particular drug, its known targets, its mechanism of action, and also get information from clinicaltrials.gov and see whether or not that drug is included in a trial, what phase of the trial it is. If it's already in the market, you'll actually see the label name for the drug and so on. So you can really dive deeply into your findings in terms of pharmacogenetics. If you were not doing a simple tumor normal matched analysis or family analysis, but were interested in looking at statistical associations, so for example, you want to see, is there an enrichment of rare variants in a particular gene or pathway in all my cases, or alternately in all my controls, or you have a quantitative trait and you want to see if there is a rare burden associated with that, rare variant burden associated with that quantitative trait, you can use the statistical association filter. Here you can set up your analysis at the gene level or pathway level. And also, you can set up your analysis to look at only case burden, only control burden, or apply a bidirectional test. We use the SCAT-O algorithm to calculate a case or control burden by keeping the directionality of the variant in mind. Also, you can run a quantitative trait analysis if you have a set of clinical features that you've included that are variable across your samples. You can use those features and covariates to run a quantitative trait. In some cases, you may be interested in doing a variant level association test, so similar to a GWAS study. You can set your analysis up to include more common variants or see if there is, for some reason, enrichment of less common variants in your cases versus controls. And so for that, you would apply our allelic association test that is here at the bottom level of the filter. You can run this test as a basic allelic model, or you can test for dominant or recessive allelic models. You can also do multiple testing corrections using either the Bonferroni or the Benjamini-Hochberg test. In addition, you can look for odds ratio, so you can see if a particular variant has a higher odds ratio for susceptibility or causing disease in your cases versus controls. What I did not speak about, oh, sorry, let's go over the results of the statistical association filter quickly. So when you apply a statistical association test, now here in this analysis, I had a very small group of cases or controls, so you will not see anything highly significant or any samples that have a large enrichment of rare variants, but you can see here that MSH2 gene here has 5 samples. Each of these have a different rare variant, but that variant is in the same gene. So this is sort of indicating that it's likely that there is something to do with the MSH2 gene that may be interesting for your study. And by clicking on the Genes tab, you can actually go in, look at all the genes that were included in your statistical association test, and also see the burden p-value. You can see which variants, how many cases and controls have the gene, so you can make more informed decisions about whether or not this is a significant finding that you would like to include in your analysis. You can also run these tests at the pathway level, and by running a pathway burden test, you can see burden p-values for the pathways. We also give you pathway association values. So for example, if you filter your genes for all the predicted deleterious variants, now you want to see, are these deleterious variants in my cases more likely to be all found in the same pathway? You can just go click on the pathway tabs. You get a Fisher's exact p-value for that pathway association, and then you can see in the pathway where the variants or genes carrying the variants are. So here we only have one gene, the MSH2 gene, that's in the mismatch repair pathway, and you can see how many cases versus controls have the variants. If you hover over these pie charts, it will give you the exact number of cases and controls that have the variant. You can actually also see that here. If there were multiple genes in the same pathway, you will see them all listed here. And by clicking on the gene name, you can see variants in that specific gene distributed by case and control. In addition to these more commonly used filters for your analyses, there are other tools available in the application to do custom annotations. So if you have a certain criteria that you would like to include at the variant level, so you have specific annotations for variants that you either included in the info field of your VCF file, or you can upload these as a bed file, you can then use that to filter variants in the application using the custom annotation filter. If you see already have a chromosomal region linked to your data and would like to look only in that region, you can type in that region in the physical location filter and then focus on that region only. You can also create lists of variant using specific filter criteria and then save this variant list and use it in other analyses for filtering variants and so on.